April 29th, we celebrate International Immunology Day. Every year, an important area of immunology is chosen as a major focus. This year, the focus is immunity through the ages, navigating the science of aging and immunology. And it is my pleasure to highlight an area of aging and immunology that I find particularly fascinating. Our immunity comes in two forms: innate immunity and adaptive immunity. Together, these provide a highly effective defense against pathogens that continually invade our body and can also prevent development of other diseases, such as cancer. Our innate immunity provides protection against a broad range of microbes and other pathogens. Our adaptive immunity differs from innate immunity in many important aspects. Adaptive immunity provides highly specific protection against the specific pathogens we encounter in our lives. Also, as the name suggests, it can adapt throughout our lives. Every one of us will develop a unique adaptive immune system, and the uniqueness depends on the specific genes we are born with, the life we live, the pathogens we encounter, and the vaccine protection we may receive. Adaptive immunity relies upon two main specialist immune cells. These are called B cells and T cells. B cells produce antibodies. Antibodies can protect us from many kinds of infections. T cells are also essential for protection from infections, and they also protect us from overactive immunity. Left unchecked, may develop into autoimmune diseases. They can also protect us from cancer. T cells get their name from the gland in which they were discovered, the thymus gland. The history of our understanding of the role of the thymus that plays in adaptive immunity is absolutely fascinating. It was first discovered, or described, to be more accurate, by Galen of Pergamon around 1,800 years ago, and is named after the Greek word for soul, or center for life. The thymus is a relatively small, two-lobed organ that lies just beneath the upper part of our breastbone in the middle of our chest. Ancient remedies and alternative medicines describe the thymus as a center of energy flow, and often practiced a technique known as the thymus thump, which they thought assisted in neutralizing negative energy, exuding calm, revamping energy, and very interestingly, boosting immunity. In 1961, however, work by Jacques Miller scientifically proved that the thymus is a vital organ or gland required for T-cell development. This actually overturned the major scientific consensus of the time that the thymus was an evolutionary accident of no great significance, and its only remaining function in us today was to act as a graveyard for dying lymphocytes, those B cells and T cells. So the T cells that leave our thymus are called naive T cells, and they function by recognizing very small parts of proteins that are presented to them for inspection via proteins we call human leukocyte antigens, or HLAs. The small parts of the proteins are generated by cutting up whole proteins from pathogens or cancer cells or even normal cells. Many of the T cells we generate in our life never encounter the small part of the protein that they can recognize. Those that do find their specific small protein in the right place at the right time can make thousands of clones of themselves. We call these effector T cells, and these cells work together to protect us from pathogen or cells that contain the whole protein from which the small parts of the protein were derived. Also, and very importantly, when these effector T cells have finished protecting us, some of them are actually kept alive. And we now refer to those cells as memory T cells. If we encounter the same whole pathogen again, these memory T cells can be awakened, can make more primed effector T cells, and these cells can mount a faster and usually better response against the pathogen or the cell that contained the whole protein. And hence, this phenomena can give us better protection than the first time we encountered the protein and pathogen. This is actually the basis of how vaccination works. Unfortunately, as we age, the diversity of our naive T cells and their ability to respond steadily decreases. It perhaps does not come as a surprise then that as we age, we become less able to fight against new infections, are more prone to developing certain autoimmune diseases such as rheumatoid arthritis, and are more prone to developing cancer. As we discussed before, our T cells are generated in the thymus. When we are born, our thymus is relatively large, about 40 grams in weight, and has two main functional areas called the cortex and the medulla. Up to around the age of puberty, our thymus is very active, generating millions upon millions of new T cells that once they leave the thymus are able to function in the rest of our body. If appropriately activated. In addition, the thymus is very good at creating molecules called thymosins. These have very important and interesting immune modulatory functions. And interestingly, we can also manufacture some of these in the lab today. And some of them are utilized to help treat cancer patients and help fight some viral infections, including COVID-19. Unfortunately, after puberty, our thymus starts to steadily lose its activity in functional areas, and these are replaced by fatty tissue. We call this phenomenon age-related thymic involution. This decline is the fastest age-related loss of activity of any organ or gland in our bodies. In fact, when we reach about the age of 65, it's estimated that our thymus has lost about 90% of the activity that it had at its peak when in puberty. As discussed, this age-related thymic involution is actually a natural process, but we do know some things that can actually increase thymic involution. These include smoking, alcohol consumption, some viral infections, and the levels of sex-determining hormones that circulate in our body. And certain cancer therapies can also induce this phenomenon, such as radiation therapy. In some people, the thymus can recover from some of the damage caused by these agents. And there is active research into how this occurs and methods we might be able to use to help the thymus recover. In fact, very recently, scientists were able to generate a synthetic human thymus. This required very clever gene manipulation techniques, special cell culture techniques, and very hard and dedicated work. It should be worth the effort, though, as we may now have a good model to test and develop regenerative methods that we can apply to ourselves and maybe even help reduce or counter the act of age-related thymic decline in activity. In the meantime, we also know aspects that decrease this thymic involution. These include vaccination, avoidance of certain excessive stress, especially stress induced by a molecule called cortisol, and following a healthy antioxidant-rich diet. I hope you enjoyed that short chat today, and thank you for listening. And if I could close with one remark, it would probably be look after your thymus and T cells, and they will look after you.